So let's get started. Good morning, everyone. Welcome to Grand Rounds. My name is Alex Harris, and I'm one of the Grand Rounds co-directors, along with Drs. Aditya Lati and Rebecca Muley. Next week's Grand Rounds will be given in person by Dr. David Pagliaccio, associate professor in our department. The title of his talk is Youth Depression, Suicide, and Links to Smartphone Usage. Before we introduce today's speaker, a reminder that if you're participating via Zoom, please post questions using the Q&A feature, and as the online moderator, Dr. Talati, will forward the questions to me for our speaker to address. We encourage trainees to ask a question, and we'll have space for trainee questions in person and online. So please indicate if you are a trainee in the Q&A. Please also remember to fill out your CME surveys after the talk if you would like credit for today's lecture. There's a QR code outside the table for those in person, and the link will be posted in the chat on Zoom at 12:15 p.m. It's now my great privilege to introduce today's speaker, a role model for myself and my fellow psychiatric neuroscientists, Dr. Bill Carlezon. Dr. Carlezon did his graduate work at Concordia University in Montreal, followed by a postdoctoral fellowship at Yale with Eric Nestler, where he pioneered one of the foundational techniques of systems neuroscience using viral-mediated transfer to show that Krebs levels in the nucleus accumbens change reward-seeking and other complex behaviors. Am I okay? In 1998, he started his own lab at Harvard, where he has been ever since, and he is currently professor of psychiatry and neuroscience at Harvard Medical School and the Phyllis and Jerome Lyle Rappaport Chair in Psychiatry at McLean Hospital. His lab focuses on the effects of stress on the brain using sophisticated animal models and a variety of systems neuroscience tools. This research has led to the development of many widely adopted paradigms and the discovery, among other things, of kappa opioid receptor antagonists, which are being tested in clinical trials as a treatment of depression. As you can tell, his work has been very influential, heavily cited, with over 225 papers. Dr. Carlezon has received several awards for his research, including the Presidential Early Career Award for Scientists and Engineers from George W. Bush, the Jacob P. Waletsky Award for Innovative Research in Drug Addiction and Alcoholism from the Society for Neuroscience, and the Daniel H. Efron Award from the American College of Neuropsychopharmacology. He's also a leader of our field, for example, as editor-in-chief of Neuropsychopharmacology and the founding editor of the new journal, NPP Digital Psychiatry and Neuroscience, and is the current president of ACNP. The title of his talk today is Preclinical Studies of Stress and Depression, Enhancing Translational Relevance. Please join me in giving him a warm welcome.
Thank you, Alex. Thank you for the kind introduction, the kind words. And I wanted to start by saying that it's a great honor to be invited to speak at an institution and before a department that's made such important and fundamental contributions to our understanding of the brain and the neurobiology of psychiatric illness. I'm grateful for the opportunity to be here today, talk about some of our work. Starting with disclosures, I want to disclose that I actually have a patent covering the use of kappa opioid receptor antagonists for treating depression. It was awarded in 2003, and it's currently expired and unlicensed. It's never licensed. So that's kind of a life lesson on how long it takes to develop these new treatments. And then I have listed some other potential conflicts of interest with other companies that I've been working with. Today's learning objectives, if you attend the session today, you'll be able to have a better understanding of how stress affects brain opioid systems, how stress affects behaviors, including sleep, and some of the animal models that study stress in the laboratory. Brief outline. I've been asked by my hosts, Drs. Harris, Talati, and Muley, to speak about our work on kappa opioid receptor antagonists, which Alex mentioned are in advanced clinical trials as a potential new treatment for depression. This is an almost 30-year journey that starts with research on Krebs. Our studies on Krebs connect us to dynorphin, which is the endogenous opioid that acts at kappa receptors in the brain. It'd be a lot easier if dynorphin acted at dynorphin receptors, but one thing we have to remember is that dynorphin acts at kappa receptors, and that's the endogenous agonist for kappa opioid receptors. Krebs I'll also be talking about translationally relevant endpoints, other cell types, other types of sleep studies we've been doing, and end up with some take-home messages at the end. So again, we're starting, this research starts with CREB. CREB is cyclic AMP response element binding protein, and it belongs to a class of molecules called transcription factors, which regulate the expression of genes. CREB in particular plays a really well-characterized role in translating events that occur on the surface up here at receptors into changes that occur in the nucleus and alter cell function, allowing the cell to adapt to experience. So CREB regulates thousands of genes, and we've listed a small handful of them here that are familiar to us, and they're genes that we've been studying in the lab. The process goes something like this. Dopamine might act, using dopamine as an example, it would act at a dopamine receptor, like a D1 receptor, and it would stimulate a cascade of intracellular changes that result in changes in CREB. So three things, in order for CREB to become activated and start transcribing genes, three things have to happen. It has to dimerize, so it finds a partner, another CREB partner. It has to sit on sites on the gene, which are called CREs, and it has to be phosphorylated. And when those three things occur, that's when you get changes in gene expression. I want to point that out because there's a way we can break CREB through a process called inducing a dominant negative mutation, where it can still dimerize, it can still sit at those sites on the DNA, but because it can't be phosphorylated, it doesn't work. And it's still sitting at the site, so it's acting kind of as an endogenous antagonist of normal CREB from getting at those sites. And that's one way we can effectively turn off CREB function by inducing this so-called dominant negative mutation. So these studies started back in the mid-90s when I was a postdoc in the Nessler lab at Yale. And one of the things I was doing was giving rats chronic exposure to drugs like morphine and cocaine and amphetamine, and then dissecting the brain and looking at how these treatments, these chronic treatments, were affecting plasticity in the brain, with particular focus on, in my case, on how CREB was changing. And we were finding that these drugs were changing CREB function in brain areas, including the nucleus accumbens, which is an important brain area for motivation. And one of the questions I was coming up with as a postdoc is, are these changes in CREB function altering sensitivity of the animal to reward? Are they altering sensitivity to stress? I see Simon there. Is everything okay? Okay, great. Thank you. At the time, there was no way to go into a brain area and to increase and decrease the function of a molecule or a transcription factor like CREB. So as Alex alluded to in his introduction, we came up with our own way. And the way we came up with was to use viral vectors. So some of the work I'm going to describe today is the first work actually involving, or among the first work involving viral mediated gene transfer in vivo to study complex behavior. And while this technique serves as the basis for many next generation techniques like optogenetics and chemogenetics and gene therapy, at the time it was highly experimental. I used to have to dress up in a spacesuit in order to do this. And in fact, when I first presented it at these data at the SFN conference, I think it was 1996, there were not a single other poster involving viral mediated gene transfer. So it was really new at the time. We weren't sure if it was going to work, how it was going to work, if it was going to make me sick, if it was going to make the animal sick. So we've come a long way in the past few decades. So here's what we found when we elevated CREB in the nucleus accumbens using a behavioral model called conditioned place preference, where the animals, if you give the animal a rewarding treatment like cocaine or morphine and pair it with a particular environment, with repeated pairings, the animal will come to approach and remain in contact with that environment and spend more time in that environment if given a free choice. So it's a very simple conditioning paradigm. If you can focus here first on these doses of cocaine that we were giving the animals, what you can see is that at these low doses in a normal animal, control animal, these low doses don't do anything. And when you get to a dose of 5 milligrams of cocaine, that's when the animals start to develop and express place preferences, and they're spending more time in environments where they previously received cocaine. So what we found when we elevated Krebs function in the nucleus accumbens was something really surprising to us. And I'll describe why it was surprising in a moment. We found that low doses of cocaine became aversive to the animals, and they were actually avoiding environments associated with this really low dose of cocaine. Then we went to higher doses. We found that they didn't seem as aversive anymore, but it wasn't inducing a normal reward type response. And when we went in and used our broken version of Krebs that actually acts like a Krebs antagonist, expressed this in the nucleus accumbens instead, what we saw is kind of the mirror image. So by blocking Krebs in the nucleus accumbens, we saw kind of immediate maximal drug responses or reward responses to intermediate doses of cocaine and still high responses. But again, I think we're at a ceiling here to the higher dose of cocaine. So the way we interpreted these data with Krebs was that Krebs was inducing kind of a combination of dysphoria at the lower doses and anhedonia at the higher doses. And of course, these are key symptoms of depressive illness. We were surprised because we thought we made the kind of the easy assumption that if drugs are causing elevations in Krebs and these drugs are sensitizing, animals are becoming more sensitive to them over time and with repeated administration. It was an easy assumption to make that elevations in Krebs would translate to elevations in reward sensitivity. And again, if anything, we saw the opposite. We saw increases to aversive properties and less reward. So we're also studying in parallel some of the things that elevating Krebs would do to gene expression in the cell. And over here, what we see is using a really kind of now a prehistoric technique. We used to have to do these one at a time. It's a northern blot that indicates that when we elevate Krebs function in the nucleus accumbens, we get more expression of dynorphin, which is exactly what we would expect, but we had to validate that this is in fact what's happening when we're overexpressing Krebs. And when we block Krebs function with the dominant negative form of Krebs, the mKrebs, we get an elimination of the dynorphin or a much reduced dynorphin signal using this northern blot. So when we moved to my lab, when I moved to McLean and started my own lab, I set up a bunch of easy and inexpensive behavioral assays, including the forced swim test, which is a stress-based assay that can identify with drugs resembling the effects of standard antidepressants. So animals treated with standard antidepressants in this assay tend to persist longer in their attempts to escape from this inescapable swim. So that would be this animal on the left. It's continuing to swim and climb and try to escape the water in this tube. Whereas untreated animals or control animals quickly develop this learned helplessness behavior and will basically stop trying to escape and just become immobile in this task. And again, this is conceptualized as a learned helplessness behavior that may be relevant to human conditions like depression. So one of the things we did was to try to understand whether this type of stress would elevate Krebs in the nucleus accumbens, and in fact, it does. importantly, we also looked at the caudate, which is a more motor area of the striatum, to kind of eliminate the possibility that what we were seeing was just a manifestation of increased behavior or increased activity in the animals as they struggle to escape from the forced swim. And we didn't see any effects on Krebs in the caudate. It was very localized to the nucleus accumbens and, in fact, the nucleus accumbens shell. So this form of stress activates Krebs in the nucleus accumbens with some degree of specificity. So what we did was we went into the nucleus accumbens and used the same approach. We either elevated Krebs function in the accumbens or blocked Krebs function in the accumbens with our dominant negative mutation. And this additional control group is just the expression of beta-galactosidase, which is an irrelevant protein that doesn't have any effect on the cell. But it was important to express some kind of new protein as the proper control. So what we saw here is pretty consistent with what we had found in the Nessler lab with drugs, with cocaine, is that elevating Krebs in the nucleus accumbens kind of reduced the latency to immobility. So what that translates to is facilitated learn helplessness behavior in this assay. So the animals were becoming immobile much more quickly when we elevated Krebs in the nucleus accumbens. That's the opposite effect that an antidepressant would have in this test. When we block Krebs function, on the other hand, just by elevating expression of dominant negative mutation Krebs in the nucleus accumbens, we saw the opposite effect. We saw greatly persistent behavior or resistance to immobility. And again, this is exactly what we would expect to see if we gave an antidepressant drug like fluoxetine or dizipramine in this particular assay. So enhancing Krebs is causing a depressive-like phenotype. Blocking Krebs is causing an antidepressant-like phenotype. So the graph on the right, what we did is we examined whether this could be due to elevations in dynorphin, which we think is the relevant gene that Krebs is regulating. So we used a drug called NorBNI, which is kind of our standard or the prototypical kappa antagonist. And we injected that into the, ICV directly into the ventricle because the drug doesn't have great bioavailability. And what we saw was evidence that this treatment, this blocking kappa receptors, so blocking the effects of dynorphin, produced a nice antidepressant-like effect on its own in control animals, and it also really strongly blocked the effects of elevating Krebs in the nucleus accumbens. So all of this is tying together really the idea that stress is activating Krebs in the nucleus accumbens, that's elevating dynorphin levels, and it's that elevation of dynorphin levels that's causing the depressive behavior. And if we can block that with kappa antagonists, we can get a nice antidepressant response in this model. So again, this led to our applying for a patent, and we received the patent. It was awarded in 2023. I mention this only because that was 22 years ago, which illustrates how long the drug development process can take, even under the best of circumstances. So I'm going to tell you a lot more studies that all look really favorable with kappa antagonists. Under the best of circumstances, it's still taking decades to develop new treatments upon identifying a new target. And this is ironic or coincidental because many of us write into our grant applications that our work will identify or provide new targets for new therapeutics, perhaps not realizing just how long this journey can take. This is a multi-decade journey from identifying a new target to getting to a place where it can actually be tested clinically in humans. So over the years, we've followed up this work with many other studies looking at behavior and starting to look at mechanisms. I want to spare you showing you a whole bunch of figures and graphs because I know that we all only have a limited amount of room in our brains for all of these figures. But I can say that it's all favorable. in a couple of decades of studying these drugs, I've seen very little in our animal models that would indicate that it wouldn't be a really good antidepressant drug and anxiolytic drug. So we've shown kind of in these classic anxiolytic tests that kappa antagonists have anxiolytic effects. So this is called the elevated plus maze. We've shown it can block the development and expression of conditioned fear using tests like fear conditioning, or in this case, fear potentiated startle, which involves pairing a foot shock with a tone or another cue like a smell, and the animals develop a fear for the foot shock and a conditioned response when they hear the tone that's been associated with that. So one of the things we wanted to do as part of this process is just check in and make sure that this form of stress, which is different from the swim stress, also elevates CREB in the nucleus accumbens. So this is foot shock. This is after 10 foot shocks, which is our normal fear conditioning paradigm regimen. And again, we're seeing big increases in CREB activation in the nucleus accumbens in response to this type of foot shock. Or this type of stress. Just as an aside, the fact that stress does this so potently, that stress so potently activates CREB in the nucleus accumbens, started to make us wonder whether the way that we were doing the drug studies when I was in the Nestor lab was actually, the changes in CREB were actually reflecting kind of the drug offset and maybe the withdrawal properties, the withdrawal inducing properties of the drugs, rather than the drug effects specifically, because it seems to be stressors that are activating CREB in the nucleus accumbens. We used intracranial self-stimulation to isolate where in the brain this was all happening. So we were injecting kappa opioid antagonists into the nucleus accumbens and looking at intracranial self-stimulation behavior. In this test, what we see is elevations in brain stimulation reward thresholds, which indicates to us that if we're injecting kappa opioid antagonists into the nucleus accumbens, the rewarding brain stimulation isn't as rewarding anymore, and we have to give the animals more stimulation to induce them to press this lever. And again, that's kind of a hallmark sign of anhedonia. It is anhedonia. We've done something that reduces the rewarding properties of a very potent reward, in this case, brain stimulation. We've also used this task to figure out if we inject kappa antagonists into the nucleus accumbens, is that where the drugs are having antidepressant effects? And all of that seems true. We've looked at interactions between kappa and stress specifically. So one way you can mimic stress in animals is to give them the stress peptide CRF, and that causes a stress-like response in the animals. And in this case, we were looking at kind of a higher level cognitive deficit that's induced by stress. So we're looking at performance in this task, the 5-choice serial reaction time task in rats. And we like this task because it's an example of a task that it's based on a task that's used in humans to study attention behavior. So we're able to retrofit this particular task to animals. So we're starting to get around to studying the same endpoints as we are in humans. We see that in vehicle-treated mice, that different increasing levels of the CRF peptide disrupts performance. So the animals stop performing well in this task, this attention task. And we can block that to a certain extent by giving a prototypical kappa antagonist. So in this case, we've moved along from Norbion, which was the initial prototypical kappa antagonist, to a newer kappa antagonist called JD-TIC, which is more bioavailable. So we can give it by systemic injection instead of having to inject it into the brain. So just to take a brief break and summarize where we are, what we've discovered, we think, is that stress activates CREB, this transcription factor in the nucleus accumbens, that by virtue of elevating dynorphin, and increasing transcription of dynorphin leads to depressive behaviors. And it's leading to depressive behaviors in virtually every animal model that we have that can be used to study depressive behavior. And I just list here kind of the basic, the most important studies, I think, in putting together the pieces of this puzzle. So we've come up with kind of a schematic of how this might work. So again, this is very simplified. I'm aware that the nucleus accumbens is more than one neuron in the ventral tegmental area. The midbrain dopamine system involves more than one neuron, but we've just oversimplified it here for the purposes of teaching. So the idea is that stress can activate Krebs in nucleus accumbens neurons. So these neurons specifically are dopamine D1, which co-express dynorphin. So we've established that we can do this through many different mechanisms, including dopamine receptors, TrkB receptors, and increasing or blocking calcium function, calcium influx into these cells. So this activates, all of these treatments will activate Krebs in these D1 dynorphin expressing neurons, and then we get elevations in dynorphin function. So dynorphin feeds back upon kappa receptors, kappa opioid receptors that are located either on the terminals of VTA dopamine neurons that are in the accumbens or in the cell bodies of these same neurons that are located in the VTA. And when that happens, kappa activation of kappa receptors by dynorphin is inhibitory. So what you get by this elevated dynorphin tone is basically decreased function of these VTA dopamine neurons. And it's known that this VTA dopamine midbrain neurons are very important for motivation and reward function. So the idea is that if we block kappa receptors, either in the VTA or in the nucleus accumbens, we can re-enable these neurons. So we go from kind of a pro-depressive or pro-anxiety state to kind of normalizing the activation of these neurons, which we think contributes to the antidepressant response. We can test this a little bit more deeply using this really unique chemical, salvinorin A. So it's a natural product. It's found in the leaves of a plant. And it's very unique because it binds, it has appreciable binding, according to the work of Brian Roth, to only one receptor in the brain, and that's kappa receptor. So we've got this drug that's very unique. It's only activating one receptor, and we can give it systemically and start making tests in that way. So in this particular figure, again, this is from Brian Roth's lab. He's lined it up against LSD, which is a much more complicated drug, obviously, with a much more complicated binding profile. LSD is acting at a lot of serotonin receptors, which are over here. So kappa isn't really doing any of those things. It's a very it's as clean as drugs can be. So when we give salvinorin A, I should mention that we had to buy the leaves, and we purified the leaves here, and we got pure salvinorin A. We had a chemist that we hired to do this, and her work was wonderful, and we were able to purify from the original product, the original leaf, pure salvinorin A. When we give salvinorin A to animals and look at brain levels of dopamine or levels of dopamine in the nucleus accumbens, we see that at an active dose, a dose of salvinorin A that in our ICSS test is causing a significant anhedonic phenotype. At that dose, we see dramatic reductions in dopamine release in the nucleus accumbens. And this is perfectly consistent with what we think is going on with that model that I presented a moment ago. When we give non-effective doses of salvinorin A, a lower dose that doesn't do anything to behavior, we don't see anything happen to dopamine, in this case, using the brain microdialysis procedure. So again, this is tightening the connection between the kappa receptors, specifically in the accumbens, and dopamine. So I just wanted to point out, to take a break to point out that this was sort of conceptually at odds with the general thinking in the field. And the general thinking in the field at the time was that elevating Krebs function in the brain was a good thing. And we had the work of people like Ron Duman at Yale and Eric Kandel at Columbia, demonstrating the important effects of Krebs, the important role of Krebs in learning, fundamental learning, and in the antidepressant response. But all of that work is based upon studies that were occurring in the hippocampus. So we're not studying the hippocampus, we're studying the nucleus accumbens, and we're seeing a much different outcome from the same process, activation of Krebs. So I think this illustrates a general principle that actions that might be beneficial in one place or one part of the body might also be simultaneously detrimental when they're occurring in other parts of the body. And I really think that that's an important point that keeps coming up in the type of work that we do. So one of the things we wanted to do as our studies have evolved and the lab has evolved is improve our animal models, improve the animal models that we're using to make them more predictive of what can be seen in humans or what will be seen in humans. And this is from a review paper that my colleague and I, Kerry Ressler, wrote. And we came up with really 3 important ingredients of what makes a good animal model. And number 1 is the translational relevance. So that means the use of similar or identical endpoints in humans and laboratory animals. Another important thing is kind of continuous data collection. So endpoints that are measured over long periods of time, as opposed to just one brief instant. So the analogy we use is moving from snapshots to film. And the final one is objectiveness. So the data can be analyzed and using rigorous algorithms, kind of minimizing handling of the animals directly or visual scoring or kind of subjective judgments about what an animal is doing or feeling based upon our understanding or of how humans might feel in the same circumstance. So you can see that some of these procedures that I've described already and that the field depends on heavily kind of fail all 3 of these criteria. And I want to add, I've used these techniques in my lab. I used them in a previous life when I worked in the primary screening laboratories for a drug company. So these procedures are pretty good at what they do. They identify standard antidepressants. They identify and drugs that have effects that resemble standard antidepressants. They identify standard anxiolytic drugs, and they identify drugs that have effects like standard anxiolytic drugs. But a lot of them, the translational relevance is questionable. Humans don't have a tail, so we can't suspend a human from their tail and make a judgment about their behavior and how they're feeling. A lot of them require kind of subjective judgments about what an animal may be doing or feeling at the time, although that has been improved by some of these motion sequencing programs that we're all using these days. And most of them require really a brief snapshot. So these are judgments about an animal's behavior that are being made in the time span of a moment or 2 minutes or 5 minutes. So they're really not continuous. So the analogy I use here when teaching this concept is that if we went to a cardiologist, it's kind of analogous to a cardiologist looking at one heartbeat and making a judgment about our health. So in reality, what happens is that cardiologists look at things over continuous data collection. So these days, if you go to a cardiologist, they may send you home with a device and they'll look at your heart data for weeks or months even before they make a judgment. So I think we need to use that analogy and move our behavioral testing procedures to better capture kind of what may happen over long periods of time instead of being so focused on being lucky enough to see an effect within a very specific and arbitrary, often arbitrary time frame. So one of the ways that we've been approaching this is to study sleep in humans and in animals. So when we're studying sleep in humans, there's a couple ways to do it. In the old days, and still presently, but in the older days, we depended upon kind of lab EEG recordings. And I want to emphasize that to get really deep insight into sleep, you need EEG. The way that we're doing it more recently is using wearable devices like our phones or smartwatches, and they can give us some insight and increasingly better insight on sleep. But they're very dependent upon actigraphy and making judgments about what you're doing on the basis of how active you are, whereas EEG is using the brain signals to come to a much more sophisticated analysis of what vigilant state you might be in. So the technique we use in our mice is kind of combines both. It's almost like a wearable, except it's subcutaneous, but we're getting EEG data and EMG data to make judgments about whether an animal is awake or asleep or what stage of sleep an animal might be in. So we can do this on a large scale. I think we have 32 of these boxes, so we can study a whole bunch of mice kind of in parallel. And I should mention we subcutaneously implant these devices which run on a battery, and they give us EEG and EMG, so muscle movement, and they also give us kind of locomotor activity and circadian rhythms of locomotor activity and circadian rhythms of body temperature, which also turn out to be important in psychiatric illness. So one of the things about sleep that we like is that it's defined and measured in the same way across species. So in humans, we can measure wakefulness using EEG and eye movement, and we're looking for things like alpha waves and beta waves, and that's telling us that the human is awake. In rodents, we do that. We can't measure, or we don't, in my lab, measure eye movement. So we depend upon EMG, and the EMG we're getting from the trapezius muscle in the back of the neck. So we're making judgments about whether an animal is awake or asleep on the basis of EMG and EMG, but we're using the same fundamental measurements to make that judgment with respect to alpha waves and beta waves and gamma waves. Slow wave sleep, also known as non-REM sleep, looks different in a human, and it looks different from wakefulness, and it looks different in a mouse from wakefulness. And REM, we call it REM. We've been encouraged by reviewers to call it REM, but in our case, when we study it in mice, it really is something called paradoxical sleep, because we're not measuring rapid eye movement in our mice. So we really should be calling it paradoxical sleep, but that term turns out to be confusing oftentimes for our reviewers. So it's called paradoxical sleep because the EEG has some features that resemble wakefulness, but there's no muscle movement at all, which is a characteristic feature of REM. So these are the ways that we use this system to tell us what phase of vigilance a mouse might be in. So why do we study sleep? Many psychiatric conditions include normal sleep or abnormal sleep, sorry, as a symptom or as a diagnostic criteria. So virtually every psychiatric illness, there's something going on with sleep. So this kind of fits an RDoC criteria, where if we're studying sleep, we're actually able to study a feature that is shared, or disrupted sleep, a feature that is shared by many psychiatric illnesses, as opposed to saying we're just studying depression or a condition like that. So stress is known to disrupt sleep. Individuals with stress-related disorders often display irregular sleep. Sleep patterns or sleep problems exacerbate other symptoms of these disorders. including insufficient sleep is known to impair cognitive function and mood. And we think that an improved understanding of how sleep interacts with stress and trauma will help progress treatments for stress and trauma-related illnesses. So the way we've been studying sleep, or one way we've been studying sleep in the lab, is combining it with this chronic social defeat paradigm, which is essentially like a bullying paradigm. It's a model of bullying, or one way to conceptualize it is it's a model of bullying, where we put kind of our target mouse, we call it, together with a larger aggressive mouse, and we allow a physical interaction that basically involves a lot of wrestling, and it ultimately ends with the larger aggressor mouse basically pinning the smaller target mouse and establishing dominance over that mouse. When that happens, what we do is we then separate the mice using a barrier, but the mice can still see each other, smell each other, hear each other. They just can't interact anymore. And for the rest of the day, we allow that kind of sensory interaction. So one way to conceptualize it, it involves kind of the physical as well as the psychological consequences of stress, like bullying. So we do this kind of in real time, and we can study the animals during this chronic social defeat paradigm, which we normally let go for 10 days. So just to familiarize you with what these figures look like, the first 5 days are kind of baseline sleep. And in this case, we're looking at REM sleep, or paradoxical sleep. These are the 10 experimental days. So we're watching what emerges with each defeat session. And then we go back to, after terminating the stress, we go back to studying the animals kind of when they're living alone, and watching how they recover from the stress. So in this case, if we look at time spent in REM sleep or paradoxical sleep, we can see kind of a rapidly accumulating effect of exposure to this type of stressor. But it seems to pretty rapidly recover when we separate the mice and the stressor ends. So that's depicted over here in the bar graph. Here's what we look at. Here's what we see when we look at, instead of looking at time in paradoxical sleep, we're looking at bouts. So it's going in and out of paradoxical sleep. So maybe reflecting sleep fragmentation. We see a very similar pattern. It's stable at baseline. It develops quickly when we expose the mice to the chronic social defeat procedure. But in this case, it doesn't really recover. So this looks like a relatively persistent effect, or at least we studied it as long as the batteries lasted, as long as we were able. So in the 5 days of recovery, it didn't look like it recovered very much. So this could be kind of a persistent effect of stress exposure, this increase in sleep fragmentation reflected by paradoxical sleep bouts. We also looked at slow wave sleep, which is another form of sleep that's been implicated in psychiatric illness. In this case, we see kind of a very similar emergence of increases in time spent in slow wave sleep, so more time spent sleeping. And this, although it looks like it recovers a little bit, there's always a persistent remaining effect that doesn't recover. So the effect is small, but the animal, it never seems to go back to normal, at least during the time frame we're studying it. So this could be kind of another biomarker of stress exposure, these increases in slow wave sleep time. We look at slow wave sleep bouts as another example. And in this case, we see no effect, or kind of a generalized non-specific effect. And this is probably just, it's basically living with a roommate maybe, because during this phase, even the control mice are living kind of across from a conspecific, so a sibling mouse. So we see that causes a general increase in sleep bouts that doesn't seem to be associated specifically with the stress, and that recovers quickly. I wanted to point out that we can do the same type of experimental design with ICSS thresholds. So again, if you remember, when we see ICSS thresholds go up, that indicates that the stimulation is less rewarding, and we need to give the mice more stimulation to get them to motivated to respond for the brain stimulation. So again, increases in threshold indicate to us that the mice are anhedonic in this particular paradigm. So you can see there's kind of a nice alignment between what we're seeing between sleep and anhedonia in this particular model, suggesting alignment, some degree of alignment between the two symptoms, sleep disruption and anhedonia. So moving back to kappa antagonists, if we give kappa antagonists to try to block this effect of chronic social defeat stress on sleep, we're able to some degree to block these effects of stress using kappa opioid antagonists. And sleep data, because it's continuous and we're measuring it in 10-second bins over weeks of time, we just get so much data. So this is kind of a core down way of presenting the data. But when we look at paradoxical sleepouts in this particular case at specific times during this chronic social defeat regimen, we see reductions in the degree of disruption that are occurring specifically on day 5, and they recover much more quickly in animals that have received the kappa antagonist than they do in control mice. We're also able to study their locomotor activity and their body temperature rhythms, and we find that the kappa antagonists completely block the effects, the flattening effects of this stressor on body temperature, which, by the way, is another sign of depressive illness, this flattening of daily rhythms of body temperature, another sign of depression in humans. Kappa antagonists block that effect, but they're not blocking the disruptive effects on locomotor activity, suggesting that these two endpoints, body temperature and activity, although they seem to go together a lot, you can uncouple them, and they don't seem to be completely dependent or linked to one another. So again, we've been trying to get down more into the mechanisms of this effect, the ability, the connection between sleep disruption and anhedonia and reward function and stress. We've used DREADS to target the D1 expressing neurons in the nucleus accumbens or the D2 expressing neurons in the nucleus accumbens, and looked at what activation of these specific cell types or inhibition of these specific cell types does to sleep behavior. I can boil down some very simple experiments into a take-home message that when we manipulate the activity using DREADS or chemogenetics of dopamine D1 cells in the nucleus accumbens, we see very specific effects on REM sleep. When we manipulate the activity of D2 cells in the nucleus accumbens, we see very specific effects on slow wave sleep. And it's only when you put these two together that you get kind of the full profile of what we see in social defeat. So it's an interesting idea that the D1 neurons, for whatever reason, are having specific effects on REM sleep and almost no influence on slow wave sleep. I wanted to point out that the biggest effect that we saw was when we inhibited these D1 neurons. That's when we saw the biggest change, or a stress-like change in REM sleep in the mice. So how does that fit with our existing model? What it suggests is that that GI dread activation of this population of cells can recapitulate kind of a secondary consequence of turning off these VTA dopamine neurons on a chronic basis. When we turn off these VTA dopamine neurons on a chronic basis, we get decreases in the activation or stimulation, the dopamine stimulation of these cells, and that's what we're mimicking with inhibition using DREADS. So it puts together two pieces of this puzzle that seem pretty consistent with one another. So again, what we're seeing when we manipulate this system in this way, we're seeing signs of depressive illness, kind of a complete profile of anhedonia, anxiety, and sleep dysregulation. And again, these are kind of the hallmark symptoms or diagnostic features of stress and depression conditions in humans. So I think we all acknowledge that sleep and circadian disruptions can play a strong role in our mood and motivation. I know if I'm not sleeping properly, I get anxious and may show signs of mood changes in mood. I also know if that I'm anxious about something, I'm probably not sleeping correctly. So there does seem to be kind of a circular relationship here. And I think what our studies show is that if we go in and we trigger using DREADS, a manipulation that we know is causing mood and anxiety signs in our animals on the basis of other work, we are able to create these sleep and circadian disruptions, establishing at least one side of this cause-effect relationship. So again, all of this work, and again, we're up to almost 2 decades of research on kappa antagonists and all of the good things they do, and that our findings in this domain, the domains we've been studying, and there's a whole bunch of other findings in the domain of substance use disorders, indicating that kappa antagonists look like they should have some clinical promise in people. And they have been developed by companies, so and tested in clinical studies in humans. And at least at the beginning, in the earlier phase studies, the phase 2 studies, they seem to work, and they seem to work in the way that you would expect. So this is this paper here reflects something called a fast-fail trial that was occurring at NIMH, where they're brief trials designed to give you basically a positive or negative sign that would enable you to either stop studying them, the drugs, or to move on to more advanced clinical studies. So in the first fast-fail trial, the kappa antagonists looked really good in people. And specifically, they looked like they were particularly effective in treating kind of melancholy or anhedonia. So there's been interest in whether that's a very specific symptom that is treated, whereas they may not be as effective at treating other symptoms. So that was work with a drug called from a company called Blackthorn, that's now Neumora, and Johnson & Johnson kind of also has their drug, a drug that originated at Lilly and then went to a company called Cerecor and then went to Johnson & Johnson. They're also finding some positive results in these early phase 2 clinical studies of these antagonists. They look like they would be good antidepressant drugs. However, more recent findings, and a lot of these are really super recent over the past couple months, have been less encouraging in phase 3. So Neumora was doing I think basically a three site study, and they one of their sites reported back, and the data were not encouraging. So they paused the studies on their drugs. And very recently, we heard the same type of message from Johnson & Johnson with their kappa antagonists. So all of this is really discouraging, particularly in light of the fact that those of us who use these animal models like to think that all of our work that we're doing can be predictive in some way of outcomes that will be seen in humans. And again, this really kind of gets, it's the old story of the preclinical studies looking really strong, and then when you get to the studies in humans, something happens and the drugs fail. So one of the things I've been trying to think about is why. Have we ever seen anything in our preclinical studies that might give us a hint as to why these drugs might be failing? And the answer is, over 20 plus years, I can only think of really 2 things that I've ever seen that would indicate in any way that these drugs wouldn't be effective for depressive-like symptoms. So in the first study I'll tell you about, we were using the chronic social defeat method to study the effects of ablating kappa receptors from specific neurons in the brain using mutations, or kappa antagonists given systemically to block these effects specifically on ICSS thresholds, so specifically on anhedonia. What you can see here in the left figure, so these are mice where what we've done is we've deleted kappa receptors from neurons in the brain that are co-expressing dopamine uptake transporter. So this is targeting the VTA dopamine, midbrain dopamine system. And when we make that manipulation, when we remove the kappa receptors from that cell type, what we see is what we would expect in the ICSS paradigm. So the chronic defeat, these mice seem less sensitive to chronic defeat, as you might expect if you were blocking the stress response in these mice. So the mutant mice that don't have these kappa receptors on the dopamine cells take longer periods of time to develop this anhedonia-like phenotype in this test. Although they ultimately get there with enough chronic social defeat, but the response is delayed. So those studies worked out the way we thought. What didn't work out the way we thought is kind of parallel studies where we gave the animals kappa antagonists. In this case, it's that drug again, JD-TIC, which we can give systemically. JD-TIC, which again is now hitting all receptors, all kappa receptors, wherever they are in the brain and the body, did not have any effect, did not have any mitigating effect on this form of stress. So it makes us wonder what these differences might be. Why this drug only works why this drug, isn't working when it's in combination with this particular task. What is unique about this particular task that might make this drug not work? And one of the things that's unique about this task, as opposed to all the others, is it does involve some inflammatory processes. So as part of the process, when these mice are interacting physically, they're often nipping at each other and biting each other. And we're removing mice from the study that really bite and cause lasting damage. But it's easy to conceptualize that this particular task, it may be particularly pro-inflammatory in the mice. And what this data set implies is that something left over somewhere where these kappa receptors are is having a counteractive or a kind of an opposing effect on depression, where when you block them, you're getting kind of a pro-depressant effect that's canceling out the effect on neurons. The other thing that we found over the years is that that kappa antagonists, and again, we're using this prototypical kappa antagonist JD-TIC, can really cause strong enhancement of inflammatory responses. And it's really interesting, and these are blood levels of these inflammatory cytokines. It's interesting that this doesn't occur kind of in a normal animal or what can be considered a healthy animal. It only occurs when you give something that challenges the immune system. And in this case, we use a substance called LPS, which activates the immune system. So when you give kappa antagonists on top of an activated immune system where there's lots of pro-inflammatory processes occurring, that's where you see a big enhancement of an inflammatory response to kappa antagonists. So we wonder how this may be happening, and where we started was on microglia, looking at microglia, which express kappa receptors. And what we did is we basically dissected out the nucleus accumbens, and this is a way of portraying kind of all of the cells in the nucleus accumbens and on the basis of the genes they're expressing, and grouping them according to similarities and differences. So that's what this looks like. This cluster is D1, this is D2. I want to call your attention to this cluster here in the middle, microglia. So when we look at microglia and specifically at kappa receptors on microglia, we see that there is some expression of kappa receptors on microglia in the nucleus accumbens. So we wondered, if this might be if we might be able to model this in our systems. So the idea would be that kappa receptors, when they're expressed on microglia, are inhibiting inflammatory processes and the release of inflammatory cytokines. So when we block those receptors on the microglia, what we're doing is kind of enhancing the inflammatory response. I'm being sensitive to time. I'll take you through very quickly these studies where we separated out neurons and microglia, and we've been studying a lot of the response in the brain. And what we find is completely consistent with the idea that that this is the way it's working, that there are kappa receptors expressed on microglia. And when you block those kappa receptors, we get enhancements in inflammatory responses, and these are specifically tied to microglia cells, or to kappa receptors that are on microglia, with a minimal contribution of kappa receptors on neurons. So of course there's this ongoing story about the role of inflammation in psychiatric illness, specifically with respect to depression. So it may not be surprising that if we're giving a treatment that is enhancing as kind of a side effect these inflammatory responses, we may be complicating the antidepressant signal that we'd love to be seeing in our humans. So one thought is that we may be want to be more attentive to the populations of people that we're studying, and if there's a way to avoid including people with ongoing inflammatory responses in these studies, that might be something we'd love to explore. So again, we thought we had figured out the mechanism on the basis of what's going on in neurons, and we had a strong mechanistic hypothesis. What we may not have been calculating is kind of the added or the side effect. What we haven't been considering more carefully enough is this added side effect that when we're blocking kappa receptors on microglia in the brain, we're enhancing inflammatory responses. So I have a few more minutes here, and this is kind of a separate little module. I know that I'm over time by a few minutes, so should I continue or should I take a break and for questions here?
Why don't we start with some questions?
Okay. Let me just scroll ahead to the take-home messages then. So take-home messages is that stress causes neuroadaptations in brain opioid systems. Stress can enhance the ability of kappa opioid receptor systems to serve as a brake on brain circuits implicated in motivation. Removing that brake with kappa antagonists as antidepressant, anti-anxiety, and anti-stress effects in animal model systems. These preclinical studies have stimulated interest in kappa antagonists to treat depression, and initially favorable clinical results have been suspended. One complicating factor may be that kappa antagonists may enhance inflammatory processes that it can interfere with potentially therapeutic effects. I'm going to skip this last point, the second to last point, and processes, and this is kind of a really take-home message, a really important take-home message, processes that can be simultaneously beneficial and detrimental. Brain processes can be simultaneously beneficial and detrimental. And again, I'm thinking of activation in Krebs, activation of Krebs in brain areas like accumbens and hippocampus. So I'll stop there and apologize for going over.